Yeah, so sir, we can start.
Yes, sir. Hello, everyone. Today we'll be dealing with on the nerve and muscle pathology in neurology, what its role is, and it's all about the spotters in the EVIVA-OS. It's mainly basically got to deal with examination questions which come towards the end when we are all very tired post the clinical case presentations, post the imaging viva, post the electrophysiology viva. This is the last thing that they ask, and it is really torturous. But that is the reason why I need to cover it up very quickly, very fast, and I need to cover a lot of things, and it's all pictorial, so it's very easy for you to analyze. And if you understand what is what, some very basics, that itself will be very sufficient. So what we'll initially do with is I'll give you, as you all know, I think by now, I think all of you would have known, when I start any presentation, I begin with history. The first thing, when we begin with history, first we need to understand, what does the word biopsy mean? Biopsy is basically, bios is life, opsis is sight or viewing. Biopsy is to view what is happening. This man was the first person to introduce or coin the term biopsy. His name was Besnier. And important to note, just historical perspective, may not be clinical exam oriented. You can let it go. As I said, throughout my presentation, I'll be giving you points where it is important, where it is not important. This is definitely not important. He coined the terms biopsy, lupus pernio, and Besnier's prurigo, which is infantile atopic dermatitis. Initially, the biopsies were done by Marcello Malfigi to begin with, who was the father of biological microscopy. Later on, we came to the current generation when there is evidence of electron microscopy, and we have two types of biopsy: excisional and incisional. In the nervous system, there are various sites we can access and view in the tissue, or we can access various sites in the nervous system: the brain, to the muscle, nerve, meninges, and skin fat, etc. These are the various sites where which gives us information. Remember, it's important to know that biopsy is just a part of the jigsaw puzzle, not the entire puzzle itself. It is sometimes the most important, sometimes not. Now, first we'll begin with nerve biopsy. What is its important role in nerve biopsy? It's helpful. We need to be very careful when its role is be very useful by selecting patients properly, selecting the nerve properly, doing the procedure skillfully and correctly, transferring the tissue specimen properly, and the reporting. We can't just rely only on lab expertise by doing mistakes in the rest of it. The lab people can help us only as much as we can help them. What is the diagnostic utility of biopsies? Initially, Western studies came as close to 10 to 24%, which help in case in deciding or diagnosing the case, whereas in Nimhan study, it was close to 37 to 39%. Now, very importantly, the next point is, what are the types of the nerve presentations, neuropathy presentations in which biopsy helps. This point is very important. Distal symmetrical neuropathies, remember, it has the lowest yield. Asymmetrical neuropathies have the highest yield. Remember these 2 points. Distal symmetrical neuropathies have the lowest yield among the biopsy clinic making the clinical diagnosis. Asymmetrical neuropathies have the highest yield. A biopsy can be of no value in close to 50% cases. It can be helpful in 20% cases, wherein it is supportive of a diagnosis, like in cases of CIDP. It is essential in 20% cases, like to differentiate true diagnosis, like for example, vasculitis versus Hansen's, and it's non-specific. This is what we usually get. Or just for the sake of doing it, there is no point. Now, please concentrate on this slide. This is very, very important. How to choose which nerve to biopsy. I used to ask this question specifically to a lot of students when they were presenting a case. The sensory nerves which we commonly pick up is the sural nerve. This is because of the proportion of the large and small fibers are roughly equal. I'll tell you what each of them means as we go on. But remember, when we have to select what type of muscle, what type of nerve, that will depend on the clinical presentation and electrophysiological involvement. Again, I tell you, do not cause an anesthetic patch in a patient just because you want to do a biopsy. Now, how do we transfer a biopsy? How do we transfer a nerve biopsy? Can somebody unmute and tell me what is the agent in which we send the biopsy sample, nerve biopsy sample? I think most of you know. Can Doctor anybody willing to volunteer? Dr. Astha, can you volunteer? What do we basically send the nerve biopsy sample in?
So one is 2.5% glutaraldehyde.
Very good. We basically send it in 2.5% glutaraldehyde, or sometimes we can send it in 10% formalin. Basically, it is to preserve the nerve in the closest biological state as it is about to be viewed. Now, this is another important thing. This is not specifically for you, but basically we need to know that there are various sections in which the nerves are processed. One is a paraffin section, semi-thin 1 micron, ultra-thin, immunostains, and teased fiber. Can anybody tell me, can somebody volunteer and tell me which is the, why a teased, where a teased fiber, how it is analyzed, and to what to do, to see what? It's okay if you can get it wrong. I'm just asking for a teased fiber.
In HNPP, sir, sausage shape, that.
Very good. No, but basically I'm trying to ask you what type of microscopy. That will come to individual cases later.
So electron microscopy.
Brilliant. Electron microscopy. Very good. Paraffin sections are for, it's the basic one. The moment we give a biopsy, what comes in 2 days is what is the paraffin section. Semi-thin sections are basically to look at axonal and myelin changes. This is what comes. I'll show it in the next slide. Ultra thin sections are for metabolic disorders. Immune stains, we all know why, but teased fiber is what they ask in the exam is for electron microscopy. Now, this is what is how a tissue is processed. Ideal length of a nerve fiber which has to be sent, please, this is a very important slide. Please remember, please note this, 2.5 centimeter. If they ask you, once 1.5 centimeter, it will be divided. The 2.5 centimeter sample ideally will be divided as 1.5, 0.5 and 0.5. 1.5 centimeter sample will ideally, among them, 1 centimeter will be taken up for the longitudinal sections. Half a centimeter will be taken up for the transverse section, and half a centimeter is for the K-Pal stain. The first initial staining, the first one which I showed you, the paraffin sections, which are the basic ones, which do come up within 1 day, are the basic longitudinal and transverse sections of the nerve, which will be analyzed, and the report will be available within a day. But the second K-Pal stains, the other stains that we use, which takes usually about 2 days, requires another half a centimeter. But the final half centimeter, for which we do a teased fiber, ideally they say teased fiber requires around 1 centimeter, but even if not, at least half a centimeter we need for an electron microscopy. Now, can somebody unmute and tell me what is the basic stains that we use? Initial staining and examination, what is the basic stains? We basically have 4 sets of stains in cases of nerve biopsy. Again, this is a very, very, very important slide. Please note this, importance of this. What is the basic stains that we know as we have studied in pathology? The most basic stains.
Hematoxylin and eosin.
Very good. Initially, eosin and hematoxylin are the basic stains. Mason's trichrome is one you need to remember. Trichrome, it has 3 colored stains. Mason trichrome is the first stain that we get. I'll show you the examples of each one of them. Can somebody tell me what are the myelin stains? The most common myelin stains that you know of? Take a guess. You can take a guess.
Paste.
Correct. 1. Okay. We have one is Kulchitsky Pal, and second is paraffin embedded stains and Luxol fast blue. Axon stains are silver stains, the Boudin silver, the palm green, the Belchovsky, the immunohistochemistry, those are all axon stains. Then we have for storage materials, which are PAS is, I don't know if somebody said PAS stain or PAL stain. It's easy. Okay. PAS stain is for, storage material like glycogen. Remember, again, this is the last point is again important. Among these two, the green part, the second one and the fourth one, you really need to remember because they'll ask you in the exam. Cressyl violet is for sulfatide storages. Congo red, as we all know, is for amyloid. Oil red O is for fat. PAS is for glycogen. I know this looks a lot, this sounds a lot like the Amish Tripathi and the books that we were reading for preparation, but this is what they ask when you have a case of peripheral nervous system, if at all, if the examiner has time. Now, very basically, a simple anatomy, the peripheral nerve consists of, the nerve fascicles. The nerve fascicles in between which are the various blood vessels. The entire nerve is covered by epineurium. The fascicles are covered by the perineurium, and the individual nerve fibers are covered by the myelin or the endoneurium. Now we have these are all continuation of the central meningeal layers. Now the most important thing is there are in the peripheral nerve. We need to know what is a node. What is a node is the junction, as we all know. I don't think I need to go back to that amount of basic. We need to know what is a node, what is an internode, the node of the nodes, the nodal gaps, the node of Ranvier, and the paranodal regions. Now, this is a typical Mason's trichrome stain. The Mason's trichrome, as we can see, has 3 colors. Okay, the 3 colors are the basic ones are the blue for collagen, and the pink or the red for the blood, and the light pink for the nerve fiber. In this, the entire nerve is seen here, as I showed you in the previous this thing. We can see here this entire section is what we are seeing here. This entire section is what we are seeing here. This blue is the epineurium, which has taken up, because of the collagen, has taken up the blue color stain. Then we have the perineurium covering the individual fascicle, and then we have the endoneurium. And of course we have, can anybody tell me what these are? Very quickly. Quickly. Anybody?
Vessels.
Very good. These are vessels. Simple. Now, Mason's trichrome, as I said, has 3 colors. Again, very quickly, for you guys to know very quickly, you need to have orientation, because the next few slides you'll understand only when you know how a normal nerve fiber looks. This is the epineurium again, this is the perineurium, and this inside is the endoneurium. Now, this is the longitudinal sections. Now, this is how a hematoxylin eosilin stain looks, and this is a Mason's trichrome stain. Now, please look at how, keep in mind, this is how a normal longitudinal section looks. These are all the axons that we are looking at, the wavy, hairy sort of pattern that it takes up. This is the longitudinal sections. This is the Mason's trichrome. Then we have the Boudin silver for the neurofilament axons. This is how, again, the longitudinal sections look. Now, I'm sorry, I thought I put the animation for this. This is the Kulchitsky Pal or the K-Pal. Okay. Now, the most important thing for this is, again, here we have the entire nerve, this thing that we are looking at, the fascicle that we are looking at. But the most important thing is the individual myelin takes up this black colored stain, and based on, we can see various sizes of the axons based on the large fiber, the small fiber, everything we can see. This is the most important slide. Please keep a snapshot of this in your minds. Okay, this is a normal nerve with a normal K-Pal staining. As I said, these are the different parts. Anyway, this is what is the large myelinated fiber, which has a very thick amount of layers of myelin, which takes up more and more of the Kapal stain. And then we have the small myelinated fibers, which have comparatively smaller ones. And then we have the unmyelinated fibers. Again, this is the type of this thing. Now, can anybody tell me this is the normal CD ratio? This is what we see for to look at or diagnose if the cup and the disc are in the normal. Then this is for the normal fundus that we see. Now, this is what is called as G ratio. This I again keep asking a lot in most of the this thing, because they keep mentioning this. G ratio is the equivalent of the CD ratio, or the cup disc size that we see in the fundus. This is the G ratio, wherein this is the axon, the actual axon or the centroid axonal diameter. And this is the myelinated fiber diameter. If we take a subtraction of this over this will give us the approximate thickness of the nerve and the myelin. Based on this, we have the G ratio, which stands for the axonal diameter divided by the myelinated or the large fiber of the entire diameter. Now we have, again, this is for the small fiber. This is the number of layers that we see, and this is the number of layers in a large fiber. Now, can somebody tell me the 7 cardinal questions to answer for a diagnosis? What all we ask in, this is again from Harrison. Can anybody tell me what are the 7 questions that we need to answer in a neuropathy case? Very quickly. Anybody can volunteer? What are the 7 questions that we need to answer? 7, I think, or 9 questions that we need to answer in a neuropathy case? Any neuropathy case? Okay. Basically, we need to answer the onset, whether it is acute or subacute or chronic, course, whether if it's rapid or slowly progressing, or whether it's relapsing or remitting, distribution, whether it's a focal disease, whether it's a multifocal disease, symmetric or asymmetric, type, whether it's a motor or sensory or mixed. Again, this is a very important slide. Please remember, please, you can take a snapshot of this at the end. The type of fiber that is involved, whether it's a large fiber or a small fiber, whether the autonomic system is involved, whether it's an axonal, demyelinating, or a mixed pattern. These are the 7 questions that we do need to answer whenever we are presenting any case of a peripheral nerve. Now, axonal reactions, what happens to injury? What is this typical thing? Whenever there is a normal axon, we have the retrograde flow, and whenever the axon breaks, this is called as the Wallerian degeneration. Wallerian degeneration is when there is a cut in the axon, the distal part of the axon breaks into pieces, and these begin to be engulfed by the local tissue macrophages, and then these small axon sprouts begin to, the small regenerating axons begin to sprout. And once they sprout, they begin to heal themselves and regenerate back into the muscle fiber. This is again what I showed in the beginning. This is the typical eosin hematoxylin staining of the longitudinal section. This is a normal appearing nerve. Can anybody tell me what these are? Very quickly. This is a normal appearing nerve. Can you make out the difference? How the axons are here, wavy, continuous, and look at what has happened to these. Can anybody tell me what this is? What stain is this? Eosin hematoxylin. Can tell me what these are? Can anybody take a guess? What shape are these? What shape is this? You can take a guess based on that. What shape is this? Can anybody take, can you guys see what I'm showing you? Can anybody tell me what shape this is? Oval, right? I'm giving you a clue. Can anybody tell me what this is? These are myelin ovoids. Okay, myelin ovoids, which mean that the axons are sprouting back. Now, can anybody tell me, again, this is another example. This is, can anybody tell me what stain this is now? Very quickly. Everyone is sleeping.
Mason trichrome.
Very good. Mason's trichrome. I think you guys, once you get a basic understanding, you'll have fun with pathology later. This is a normal stain. Now, as I said, this is again Mason's trichrome. This is how a normal nerve is supposed to look. Look at the amount of the blue color that is inside the perineurium. This is what is called as endoneurial fibrosis. When there is severely increased amount of fibrotic tissue inside the nerve, this is what is called as endoneurial fibrosis, which takes up a lot of the blue colored stain in the Mason's trichrome. This is the normal one. This is what happens when there is fiber dropout, or this again is the endoneurial fibrosis. This is what is if the nerve axon is unable to generate and it is replaced by fibrous tissue. When the axons take up the blue the pink color, the collagen takes up blue. When the axon is replaced by the fibrous tissue, this is what is called as fiber dropout or endoneurial fibrosis. Can anybody tell me what these are? What stain is this, first of all? Very quickly.
Kapal stain.
Very good. Very
Kapal
good.
stain.
Very good. Brilliant. Can anybody tell me what I have highlighted? This is the
Myelin.
tarr fiber. Very good. This is the myelin. Can you see the sprouting? These are
Yes, sir.
called as bands of Bungner. These are the axon regeneration clusters. This is what I showed you, how the axon is going to regenerate. It is going to regenerate in short sprouts. Can you see the size of the axons here and here? They are small and they are generating themselves. These are called as bands of Bungner. This is what is when there is demyelination and remyelination in the same slide. We'll come back to this. Now, basically, myelin ovoids mean there is acute myelin breakdown. This is what we see in cases of quickly.
GBS.
Very good. GBS. Very good. Come on, guys. You guys know most of it. I'm just giving you pictures of what you know. That's it. Denuded axons are what we see from semi-thin sections. Myelin thin onion bulbs represent demyelination and remyelination. Remember, myelin, if somebody asks you acute myelin breakdown, acute GBS, what do you see in, first of all, if they tell you, do you do a nerve biopsy? What is the answer? Do you do a nerve biopsy for GBS?
No.
Very good. This is all
No, sir.
theoretical. Remember, no. Myelin ovoids are, these are all theoretical, but again, if it represents, by chance, if you have a patient of CIDP who has presented with an acute GBS like episode, that is when this actually helps. Okay. Now, again, active demyelination. Can anybody tell me? Look at this. This is the myelin ovoid. Can somebody tell me what stain this is? The first one. What color is this?
Kapal.
Very good. This is simple
Yeah.
LS section, longitudinal section. This is the same myelin ovoids. Look at this. The same thing, the denuded axons. Whereas in chronic demyelination, can somebody tell me what? Oh shit! Can somebody tell me what this is?
No.
Yeah, this
Onion
is.
peel appearance.
Onion
Myelin
bulb.
myelin.
Yeah, this is how an onion bulb appearance will look when there is demyelination and remyelination occurring. Chronic demyelination, when the nerve is trying to heal itself by covering itself, there is repeated chronic remyelination. This is the typical onion bulb pattern. Okay, does it look the same? Yeah.
Yes.
Then we have the normal. Now can you make out the amount of fiber dropout? Look at this is what I have been putting from the first slide. This is the normal Kapal stain. Look at the amount of mild axonal loss, moderate axonal loss, severe axonal loss. This is what they give you in the Niemann pathology report. This is what you need to be interpreting it as. You guys can make out, right? Mild axonal loss, moderate and severe. Sorry. Yeah. Now, can anybody tell me, this is the 40-year-old adult. This is a 4-year-old child. Look at the thickness of the fibers. Now, more important than that, I'll come back to all this later. This is what happens when you do mistakes. This is the normal glutaraldehyde, which preserves the nerve fiber. If you put it in formalin, this is how the nerve fibers become. You can in no way interpret the biopsy report. Now, these are what are called as the saline artifacts, which are during biopsy. This again is not important. Now, this is significant. I think most of you guys were telling when we stretch the fiber, If the nerve begins to collapse on it, again, this is a stretch artifact. Remember, there are two sorts of artifacts that we need to remember. One is a saline artifact and the other is a stretch artifact. Can anybody tell me what this is quickly? Somebody just mentioned this, the first response that I got. Can anybody tell me what these are? What do they look like? This is a, what stain again this is this? This
K-pulse.
is what stain? This is what stain the
A
first?
trichome.
Very good. This is a trichrome LS. This is
Okay.
the K-pulse. Can anybody tell me what this looks like? Any guesses?
Hypermyelination.
Very good. One could be hypermyelination, or somebody had just told me initially, this is just a pinch type. But this looks a lot like, if anybody had told me this looks like tomatoes, I would have agreed with them. But this is a pinch artifact. Now, again, as we said, we need to see if there is inflammatory infiltrate when there is inflammation like macrophages, or if there is segmental or de or remyelination. In AIDP, again, this is not important. Again, this is definitely not important. Now, CIDP. Now, in CIDP, what do we see? This is when we send the patients for biopsy. We see inflammation in 50%, onion bulbs in 20%. Not all CIDP patients need not have onion bulbs. Onion bulbs mean, can anybody tell me what? Very quickly.
Myelination and remyelination.
Very good. Demyelination and chronic remyelination. It means that the nerve is trying to heal. If you see onion bulbs, what is the prognosis that you will give? Good or bad?
Good.
Better.
Very good. Yeah, better prognosis. Come on, guys. Simple logics. Then if there is edema or if there is demyelination, poor prognosis. Got it? See, you guys are only understanding everything. I don't have to tell you. Once you get the logic funda. This is a normal stain. This is the when we have the endoneurial inflammation. Can you see the this is the normal blood tissue macrophages that are engulfing the perivascular tissue. This is the onion bulb. This is the demyelination and the remyelination. Again, this is normal. These are the onion bulbs. This is severe demyelination when there is no nerve fibers that are seen on K-pulse. And this is the hypertrophied fascicles. Can anybody tell me where we get hypertrophied fascicles? Hypertrophied fascicles when
HMSN.
Dystrophy.
simple. Yeah, HMSN. Correct. When we have this is another way of saying nerve thickening. Thickened nerves. Correct? Thickened nerves. Correct. Very good. So in HMSN, we have hypertrophied funicles, hypertrophied fascicles. In HMSN1, we have hypertrophied fascicles. In HMSN2, we have small funicles. Remember this. Okay. Now, pseudo onion bulb patterns appear in true onion bulbs are in HMSN1. Pseudo onion bulbs are in HMSN2. Can anybody tell me what this is? I just told you.
Pinch artifact.
Maculars.
Yeah.
Pinch artifact.
No. Very good. Pinch artifact. No. Now I'm giving you the.
Macular.
Yeah, correct. This looks like a tomato. This is a typical tomaculous neuropathy, which is seen in HNPP. Correct. This is what looks like a tomato, right? Can you get it? This is what looks like a typical tomato. This is the tomaculous neuropathy. This is the, again, tomaculi. Can you make this out? Tomaculous neuropathy. This is a pinch artifact. Now look at how from center to out, look at how it looks. Actually, there is an absolute difference. Then there is, if there is no clustering or no this thing, then it's more looking towards like a pinch artifact. Okay. Now again, this is the Raynaud bodies are important for again. Remember the name. This is in compressive neuropathies. Raynaud bodies. Remember this this name, that Raynaud bodies. Just I don't think they'll ask you. Sorry. This again is when there is severe axonal dropout in cases of hereditary sensory axonal neuropathies. HSAN1, in HSAN2, HSAN3 when there is large fibers are preserved and small fibers are involved. Can you make out, if anybody can understand that all the fibers are gone in HSAN1? Look at this, all the fibers are gone. Small, large, everything is gone in HSAN1 and 2. But in HSAN3, look at the large fibers which are preserved. Again, in HSAN4, large fibers are preserved and the small fibers are gone. Okay. Can anybody tell me what this is? These are what? These are what? These are the ones.
Inflammatory cells.
Correct. Very good. Very good. These are the inflammatory cells which are surrounding the vessels. That means what is this?
Perivascular.
Correct. Perivascular, which is what does it
Inflammatory.
indicate?
Inflammatory.
Inflammatory mind.
vasculitis.
Very good. Madam, you are good to be a pathologist now. This is a typical vasculitis. Correct. Based on the type of inflammatory infiltrate, we decide what is the type of the inflammatory cell which has which has begun to involve the vessel. Based on that, we decide what is the type of the disease that we are seeing at. Look, this is the vessel wall. Look at the inflammation, that the amount of inflammation there is, and it has caused a complete necrosis of the nerve. This is the necrotizing vasculitis. Again, again, you need to look at neovascularization. These are not important for you. Again, you can let this go. Can anybody tell me what 1, 2, and 3 represents? 1 represents what? 2 represents what? 3 represents what? Very quickly. I have mentioned, I mean, it is blocked.
Epiperiendoneurium.
Yeah, exactly. The vessels in the epineurium, the vessels, vasa nervosum, the vessels in the perineurium, and the vessels in the endoneurium. Brilliant. We have basically, based on that, we classify into large vessel vasculitis or a small vessel vasculitis. Large vessel vasculitis, we have PAN, we have Wegener's. I'm sorry, we don't no longer use the term Wegener's. Can anybody tell me what is the word we use?
Microangiopathy.
Microangiitis.
Eosinophilic polyangiitis.
Is it eosinophilic polyangiitis? Can anybody tell me? Can anybody guess? Why the term Wegener's was removed?
The Nazi.
Yeah, brilliant. Then we have the epineurial vessels again. If again it indirectly begins to say that whether it's a large vessel or an endoneurial, which is a small vessel. Okay. Okay, these are the types of cells. Again, eosinophils, again, which means necrotizing vasculitis, like in Church straws, or plasma cells, which we see in PAN. Again, this is again not important for you. This I'm not sure. No. This again is not important. Yeah, this is again very important. Now, can anybody make out the difference between the two? Can anybody tell me what is what you are seeing in the second slide on the right side? This is a normal one which I'm using from a long time. Can anybody tell me what is the type of what are the type of fibers that are lost here?
Atrophy of the small fibers.
Smaller fiber.
Small and unmyelinated.
Yeah, exactly. Most normal KPAL, this is the small fiber neuropathy. Correct. Now, what are the causes of small fiber neuropathy? Diabetes, amyloidosis, leprosy, HSAN, Fabry's, Tangiers. Can anybody tell me what this is? Look at the fascicle. Look at the fascicle, type of the fascicle. What has happened? Whether do you, is this the normal fascicle that I was showing you?
No.
No.
Yes.
That means there is enlarged. The nerve appears enlarged. This is a typical leprous neuropathy, lepromatous neuropathy. Again, in lepromatous neuropathy, what are the types? What are the types of leprosy?
Lepromatous
Tuberculosis.
tuberculous.
Correct. Two zones, if you tell me, that is sufficient. What tuberculoid leprosy and lepromatous leprosy. Tuberculoid leprosy means what has happened to the immunity? Immune response is what? In tuberculosis?
Severely
Is
depressed.
Depressed is tuberculosis?
Is good. Is good.
Yes,
Immune
very
response is
good. Very good. Tuberculosis, it is good. Lepromatous, it is poor.
Bad.
Correct? So if you see a hypertrophied fascicle, if you see inflammatory cells, what type of leprosy is it? Can you see the inflammatory cells here?
Tuberculosis.
The macrophages here?
Tuberculoid leprosy.
Correct. This is a tubercular, tuberculous leprosy. This is the giant cell. This is the giant cell that we see. Got it? Again, this is the transverse, this is the lepromatous form. This is the healed. Can anybody tell me what has happened? Can happen? This is the entire fascicle.
Fibrosis.
Very good. Why is the fibrosis happening?
Blue color.
Correct. Very good. See, you guys are interpreting it so well. Brilliant. Blue color. So there is more blue color, the collagen.
Collagen.
Correct. So that means the immune response is very good. So it's a healed tuberculomatous type. Then you have the lepromatous bacilli. When you see the lepromatous bacilli, the lepra bacilli, then it is a lepromatous leprosy. Again, this is granulomas, you can let it be. Now, this is a very important stage. Can you see the squashed appearance? Can you see the squashed appearance around the vessel? This is important. Just have a look at the squashed appearance. This is what is called as a perivascular deposit. What all deposit diseases do you know? I showed you
Glycogen.
things.
Amyloidosis.
Very good.
Amyloidosis, glycogen.
Very good.
Sarcoid.
Madam, I think you need to stop answering. I think you can leave the class. You're brilliant now. I think you know a lot of pathology by now. The others need to participate. I think you guys have fun when you keep seeing, interpreting the images. Second, you have this sort of K-PAL is showing small fiber loss, isn't it? You can see the large fibers, correct? But the small fibers are the ones which are gone. Small fiber neuropathy, it's perivascular deposit. Any guesses? What I'm leading?
Amyloidosis.
Very good. Brilliant. So what is the diagnostic stain in amyloidosis?
Congo red.
And what is this? What do you eat? What is the pattern that you see, Baba? So what is isn't this like a green color that you see? Can you make out?
Apple green.
Apple green.
Brilliant. This is the apple green birefringence. Correct. This is how it looks. Are you getting my point? How do you interpret? This is the vessel. This is the perivascular this thing. You look at the K-PAL if it is showing small fiber loss. Look at if there is the on Congo red, if there is then you have the apple green birefringence. Got it? So this is amyloidosis. Very good. Very good. Can anybody tell me what this is? Come on, guys. Just looking at this thing isn't sufficient. Come on, take a guess. I'm showing you hair neuropathy. What else?
Kinky hair.
Yes.
Axonal.
Mother's normal hair, thick wire coir-like hair.
GA.
Come on, guys. More people need to participate. Irregular
Giant
eyeliner.
axonal
This is GAN. Yes. This is giant axonal neuropathy. This is the thick, wiry, coir-like hair. Remember this. These are all, everything is pictures now. That's what I said. This is not a clinical case presentation. This isn't a clinical presentation. So you guys can participate. Okay. So basically, if again, this isn't that significant, polyglucosan, need not worry. Nerve biopsies when to do, again, diagnostics and suggestive, this all I have covered. When not to do. Do you do a nerve biopsy when it's metabolic? No. Toxic. You just are not going to be able to ideally differentiate what toxic you are able to. You'll be just able to tell whether there's the type of pattern loss, axon severe or not, but you'll not be able to identify the cause. Hereditary, again, when not to do, when you know that it is this thing, again, to prove HNPP, I agree. Paraprotein paraneoplastic, again, GB syndrome, most important. Now, very quickly, are we done with nerve biopsies? Any questions you have? You guys understood what is normal K-PAL stain, how a large fiber looks, how a small fiber looks, what are the Mason's trichrome stain, how to look at particles, how to look at the vessels, how to interpret perivascular fibrosis. You guys understood that? All of it?
Yes, sir.
Brilliant.
Yes, sir.
Because now it takes on a higher level. Now it comes to the muscle biopsy. That's the reason why. Nerve biopsies are relatively easy to interpret. Now it's real fun. Muscle biopsies. Now, why do we do a muscle biopsy? When there is weakness, when we don't know the cause, when there is muscle pain. Again, when we have general reasons, specific causes. Now, this again is a very important slide. You can take a snapshot. You can remember this, mug this. I don't know, but you need to know this. Chronic diseases, which muscle to choose? When there is muscle with moderate but not severe weakness. What happens when you choose a muscle with severe weakness? There is nothing that you'll get other than fibrosis. What happens if you choose a muscle with no weakness? You'll get no result at all. We need to choose a muscle with moderate weakness in cases of a chronic disease. In acute disease, on the other hand, we need to choose a muscle which is which is showing severe weakness, because that is the pathological muscle. In best specific muscles, we need to choose the deltoid, biceps, or quadriceps based on what our diagnosis is. Ideally, we have to choose a muscle which has equal number of type 1 and type 2 fibers. If at all, if we are planning a combined biopsy like a nerve and a muscle, we go ahead with a sural and gastroc or a SPN and a peroneal muscles. Dubowitz, I think the pathologist will know. Dubowitz is one of the legends of muscle biopsy, who said a small fragmented of an unnamed muscle coiled into a disoriented ball after being dropped into formalin is unlikely to give any meaningful interpretation, no matter how careful you process it. It's of no use. Can anybody tell me the types of muscle biopsy very quickly? Pat. Very fast. Types of muscle biopsy. Okay, this isn't that important, but make sure you know it. There is a needle biopsy, a conchetome biopsy, and an open biopsy. We invariably do a open biopsy these days because, again, I know the wound healing will be slow, but it can lead to a scar. But again, open biopsy, when we combine it with the muscle and the nerve, it's ideal. Open biopsy. Remember these two words. There is a possibility of doing a needle biopsy. Remember the word conchetome. Remember it. That's it. This is the typical needle that we use. The biopsy from the biopsy site, it will be a beveled edge needle. This is called as the Bergstrom's needle. And secondly, this is the Blakel sleeve muscle biopsy conchetome. This just looks like an artery forceps. This is the conchetome. Now again, when this is the indications for muscle biopsy, the do's and don'ts. The first thing, do not drown the muscle biopsy in saline. Okay, you need to do excision. Do not do excision biopsies. You don't excise the muscle by cutting it, pinching it, or tying it. You can't tie the two ends of this thing. You need to gradually, when you take a muscle biopsy, you take a 2 by 2 centimeter, wrap it in a gauze with few drops of saline to keep the muscle moist. That's it. Do not drown it. Do not, look at this. You don't twist the muscle fiber. You don't tie the muscle fiber. You don't pull the muscle fiber. And most importantly, do not drown it in saline like this. You just need to soak it. A few drops of saline to keep the muscle moist until it reaches the lab. There is no specific sample, that actually glutaraldehyde samples are needed when you are planning for electron microscopy, but don't worry about it. Remember, fixative, just normal saline soaked gauze. Okay, you cannot delay muscle biopsies. It should reach the lab within a period of 4 hours, because if you are planning to do for a electron microscopy, if not, if for immunohistochemistry, just for the routine stains, again, ideally it has to be sent very quickly. You wrap it in a few saline soaked gauze, place it in a Ziploc bag, and then put it in a flask containing ice. Once the tissue reaches the lab, what they do is they chill it in a liquid nitrogen. They freeze the muscle. The sample is frozen at -80 degrees in isopentane, and then the sections are taken. The cryo sections are taken, and then you have 3 various ways. Again, just like the nerve, it is sent to enzyme histochemistry, to immunohistochemistries, the enzyme assays, muscle cultures, and then to electron microscopy. Basic anatomy, just like the nerve. Again, I repeat, I am not going into different clinical aspects, or I don't want to describe the patterns of the clinical weakness and all. I just want you to understand how a muscle looks, the epimysium, the perimysium, and the endomysium. The endomysium is the one which surrounds the individual fiber. Okay, we all know what the Z disc is, what the H zone is. And remember, when we know what can anybody tell me? Again, you need to remember basic 2 things: actin, myosin. The actin is the sorry, can everybody hear me?
Yes.
Yeah, anybody. This is the actin filament, which is the smaller one. The myosin filament is the large red one, which is held together in place by where the Z disc is attached by the titin. This is the titin filament, which holds the myosin fragment inside or in between the two Z discs. Again, this is all the myosin, what the troponin, the tropomyosin, and the myosin. Again, this is all you would have read about all this. This is a very important picture. This is available in Google. Please, 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 whoever is the exam going PG, exam going residents, take a snapshot of this picture, keep it, paste it on in your Bradleys. Remember this, keep studying this. This gives you an entire picture as to what happens, what are the channelopathies, what are the muscle diseases involving which protein. This covers, I think, a lot of proteins. Like, for example, the dystroglycan complex. Dystroglycan complex is associated with the dystrophins. Then we have the dystrophin proteins. Then we have the dysferlin, which is the channelopathy for, which is the, not a channelopathy, which is the LGMD2B protein, the caveolin protein, the calpains, the titins, everything. Okay, this is a very comprehensive slide. You can go for it. There are 3 types of muscle fibers. Again, the type 1, the type 2, which has 2 types, type 2A and the type 2B. For long distance running, we need the type 1 fiber. For short sprints, we require the type 2B fibers. Remember, why is it important? Because the fatigue rate is slow or the low in type 1 fibers. We require a lot of energy very quickly in type 2B fibers. So what happens to the mitochondria? Can anybody tell me the amount of mitochondria? The mitochondrial density is obviously very, even though type 1 fibers will have very high, type 2A fibers will have very high, type 2B fibers will have low amount of mitochondria. Based on that, we decide, again, as I said, there are, I'll go by the types of stains in the muscle biopsy. We have 2 types of stains. One is the enzyme stains and the non-enzyme stains. I'll be dealing with each one of them very quickly. These are the basic stains in muscle biopsy: hematoxylin and eosin for general architecture, Mason's trichrome to assess the connective tissue details. Again, muscle is red, collagen is blue, nuclei are purple. PTA stains are for ring fibers and cross striations. You can take a snapshot of this when I complete it. Congo red is for amyloid. Modified Gomori trichrome. It's not a typical Mason's trichrome. It's a modified Gomori trichrome. Is for nemaline rod, ragged red, rimmed vacuoles, and tubular aggregates. Oil redo is for lipid storage. PAS is for glycogen storage. What is normal? As we all saw, as I explained it to you earlier, the perimysium, the epimysium, which is the one which surrounds the entire muscle. Then we have the perimysium, which surrounds the fascicle, the muscle fascicle, and then we have the endomysium, which covers the muscle fiber. Got it? Now, please again have a look. Just like how you can forget, now you can forget all what I told you initially. Please just remember this. Okay, this is a normal slide. Just like I showed you the Kapal stain, this is a normal muscle biopsy slide. Okay, HNE. HNE shows this should have shown the rim is the epimysium. Then we have the perimysium here. Can you guys see the white thing is the perimysium? The individual one is the endomysium. Okay. Now this comes to the different types of fiber differentiation. First, I'm just going to show you the types of fibers. One is I'll come back to these enzyme stains later. Remember, there are 2 forms, 1 and 2. You need to remember, basically differentiate type 1 and type 2. Type 1 will appear in the SDH. Type 1 will appear darker. In the NADH, type 1 will appear darker. Type 2 will appear lighter. And in COX enzyme, in COX staining, type 1 will appear darker. Type 2 will appear lighter. Okay. Now, fiber distribution in normal type 1, which have more mitochondria, it will appear darker. Got it? Hello?
Varun, can you unmute? Can you mute the others?
Yeah, that's it. In type 2 fibers, we have fewer mitochondria, hence the lighter, it'll appear lighter in this thing, in the biopsy. Ideally, the ratio of the type 1, type 2 in quadriceps will be 1:2. In type 2 fibers, in type 2 muscle fibers, the 2A, it should ideally, in the type 2, there are 2A, 2B, and 2C, but ideally, remember, it should be 1:1. If type 1 fibers alone to say a muscle is type 1 dominant, again, it should have more than 55%. This again, just like the CIDP criteria, this is again, sometimes some examiners ask you how to say that a muscle is type 1 dominant, predominant, when there is more than 55% of the fibers should be type 1. To say it is type 2, it should be more than 80% should be type 2. Okay. This is the normal muscle when the type 1 fibers appear darker on SDH, on NADH. These are the enzyme stains. In the typical 9, this is the enzyme stain that you should remember more commonly, the 4.6, in which type 1 appears darker, type 2 appears lighter. In higher enzyme pH enzyme stains of 9.4, it is reverse. But remember these three at least: SDH, NADH, and ATPase. Okay. Now, as we can see here, for the hematoxylin eosin, can we see the amount of type 1 fiber, which is type 1 fiber dominance? Or this is, can anybody tell me what is the what is the pH of this ATPase? If this is type 1 fiber dominance?
9.4.
Very good. This is 9.4. Brilliant. Can anybody tell me what is the fiber, what is the pH of this? This is again type 1 hypoplasia. Hypoplasia. That means, again, we are looking at the where type 1 is lighter, which means it is still 9.4. Got it? Now, general patterns. Now we look at the, again, this may not be very significant, but remember these things. This is the normal section. The cryo, this is the normal one. This is how a normal muscle fiber should look when they are atrophied. Can you look at how small these fibers become? This is the atrophied one. And I'll come back to it. I'll explain when they become large, it becomes myopathic. I'll explain to you each one of them. Now, this is what is the normal fiber, and this is the necrotic fiber. When it is engulfed by the macrophages, this is the necrosis, the tissue macrophages. Wherein this is the myophagocytosis, just like the, this looks like what immediately when I showed you another picture of this. This looks like what immediately? This is a muscle, I agree. But this looks a lot like the myelin ovoids that I showed you initially.
Ovoids, yes.
Myelin ovoids that I showed you. If they look like a lot like that, it looks like just like how the myelin is being phagocytosed, the muscle is also being phagocytosed. This is myophagocytosis. When there is a lot of inflammatory infiltrate, this is myositis. And when there is regeneration, this is not that significant. Now, internalization of the nuclei. Normally, as I showed you initially, the nuclei are all peripherally placed. Why? Can anybody tell me why the nuclei are peripherally placed? What is inside each endomysium? These are all the fibers, remember. These are all the actin, myosin that we are seeing. The nuclei, the muscle, as we know, the skeletal muscle, if we can remember our basic histology, skeletal muscle, the nuclei will be peripheral. When will the nuclei become internal? This is again very important. Internalization of nuclei when there is a myotendinous insertion, or when it is very close towards the tendon, or if there is, this is normal, if the centronuclear myopathy, when the nucleus shifts towards the center, in myotonic dystrophies, in fiber regeneration, and in chronic neuropathies. Remember, okay? This is important for you, but again, remember when there is nucleus, it becomes internal. Again, this isn't that great important. Can you see where the nucleus is here? Can you guys see? This is almost like a bindi that each muscle endomysium has. Can you see this?
Yes, sir.
Yes,
See
sir.
the nuclei here. This is where it is internalized. This is the centronuclear myopathy. Is this how we remember our skeletal muscle histology? No. Normally the nuclei will be here. It will be in the periphery. When it comes towards the center, this is called as centronuclear myopathy. This is the Duchenne, wherein there is the fiber type. I'll be explaining it to you. This is the walled fiber. Can you see how this is look almost like a tomaculus or a onion bell pattern? This is what we see in LGMDs. Then we have fibrous necrosis based on where the necrosis is happening. Based on that, can we see the again longitudinal sections? You can let it go. In the transverse section itself, this is the normal fiber. This is the vascular, the vessel where which runs in the endomysium and the perimysium. This is the perimysium. Say this is a medium vessel. Can we see the necrosis that is happening around this vessel? This is called as the perifascicular, around the fascicle necrosis. Can anybody tell me what is the diagnosis in this? Perifascicular necrosis?
Dermatomyositis.
Brilliant. This is dermatomyositis. Nuclear inclusions. Can we see? I said initially the inclusions are supposed, if it is staining purple and the inclusions are inside, this is what is seen in OPMD, oculopharyngeal muscle dystrophy, OPMDs. Then we have this is again the Gomori trichome stain, wherein if there is a sarcoplasmic inclusions, which basically are seen in IVMs and in myofibrillar myopathies. This isn't that important. Again, not that important. This again shows you a picture like this. This is called as fiber splitting. Can you see this? This one band here. This is called fiber splitting, wherein the entire endomysium is split in a linear split, slit-like thing. This is our this again isn't that mottled fiber. Yeah, I mean I shouldn't have put the direct pictures here, but remember this, the mottled fiber. What can anybody tell me what stain this is? What are the initial three stains that I showed you? SDH, NADH, and
ETP, COX.
COX. Correct. Very good. If the fibers appear normally, you saw the pattern, how uniform the fibers should have been. These fibers are losing that pattern. This is what is called as mottling, mottled fiber appearance, which is seen in FSHD. Okay. Again, this isn't that. This is a rimmed vacuole. This is a freezing artifact. Again, this isn't that important. Yeah, this is what is important for you whenever this is the picture that they show you. Inflammatory myopathy, when I said there is perivascular. Can you see the macrophages here? Can you see the macrophages around the vessel? This is the perivascular endomysial inflammation, which leads to the necrosis of the muscle fiber. Look at the muscle fiber here in the fascicle, whereas towards the rim of the fascicle, the muscle fiber, can you make out the size of the fiber? Each endomysium, how small it is, just like the K-pal, how you were looking at interpreting in this thing. This is how it has become small towards the edge of the fascicle. This is what is called as perifascicular atrophy, because the antibodies are coming out of the vessels and destroying the muscle fiber around them. The problem is in the vessel, but not in the muscle. Remember that. This is the perivascular infiltrate or the perivascular fascicular atrophy, which is seen in dermatomyositis. This is the slide that is important. You can take a picture, snapshot, do what you want. Remember, normal fiber, when again I'll recap everything in this one slide. Again, you could have asked me, I could have just shown this. I'm sorry, that is the point of a lecture. This is the normal fiber when the nuclei appear distal. When they are atrophied, the fibers become smaller in size. When they become angulated, this is called as angulation of fibers. Think of denervation. If there is a vertical split, think of a dystrophy. Ringed fibers, when the individual fibers are in a rim, think of myotonic dystrophy. Target fibers you see in reinnervation, just like onion bulbing. Central core disease, when everything is when the fibers are placed inside in a small core. We know Nemeline rods. I'll show you them. I'll show you what Nemeline rods look like. Then we have myophagocytosis and fiber regeneration. Mason's trichrome, again, I'll come back to all this later. This is again what is important. This is the PTAH stain. Again, if somebody asks you why this is important, this is the typical, what did I show you for myotonic dystrophy? Ring fiber. This is what is the ring fiber. Can you see this? The ring here. This is almost like a sleeve of a ring. This is seen in myotonic dystrophy. Remember the names, it is sufficient. Ring fibers are seen in myotonic dystrophy. Then we have again myotonic dystrophy. This is another example of a ring fiber. Can you make out the difference? How this thing is? This is just like if at all in the exam, what is the EMG pattern that we see in myotonic dystrophy? EMG sound that we hear in myotonic dystrophy?
Bomb
Dive
diver.
bomber.
Dive bomber. Dive bomber. If you really, this is how I used to remember it. The dive bomber of the muscle biopsy is myotonic dystrophy. Okay. Then we have the nemaline rods that appear like this. Then this can anybody tell me what this is? CB. What does CB mean? This is the ragged red fibers which are seen in
Mitochondrial.
mitochondria. Tubular aggregates. Rimmed vacuoles are seen in
GNE.
brilliant. Lipid storage diseases, again, this is how, again, this is all not that this is all not that important, but remember again, stains you remember for the disease. Oil redo for lipid storage, PAS for glycogen. Congo red, again, this is, what is this?
Amyloid.
Amyloid.
Very good. This is the apple green birefringence. Okay. This is the enzyme stains. Remember, there are a whole host of enzymes that can be assessed because the muscle is an active tissue. We look at the enzymes coming from the mitochondria. We look at the ATP enzymes, the NADH and the SDH. Got it? These are the three stains that you remember. Remember, SDH, NADH and the ATP. These are the main enzyme stains. SDH, again, to decide whether they are the types of look at the central core disease, the target fibers, all these are not important. Remember the names, it is sufficient. NADH, SDH and ATPase. Again, ATPase, there are three. Remember, 4.6 is the very important one, which I showed you. All this you can go. Myophosphorylase again is for McArdle's. That is one thing that they might ask. But again, SDH. In SDH, it is specific for mitochondria, in which SDH, as I showed you, is a, look at this, sorry, I'm extremely sorry. SDH is inside the mitochondria. It is very specific for the mitochondria. This is the entire cell. This is the entire cell. But inside is what SDH is inside the mitochondria. NADH is outside the mitochondria. ATP is again outside the mitochondria. Got it? SDH is specific for mitochondria. More mitochondria that you have, it will be a type 1 fiber. More dense, lesser mitochondria you have, it will be a type 2 fiber. Got it? NADH again is again very specific for mitochondria and the sarcoplasmic reticulum. Again, type more mitochondria, type 1, lesser mitochondria, type 2. And cytochrome again is again very specific for mitochondrial specific enzymes. If somebody asks you what are the types of this thing, we look at basically these three. ATPase, we have three pH. One, the most important one is 4.6. Remember that is more than sufficient. Type 1 is dark, type 2 is light. I think we are going way. I think these are all significant, not that important. Again, centronuclear myopathies, I think I dealt with all this later. SMA, this is again important. Group atrophy, group hypertrophy. Okay, if you can look at the type of fascicles here, the fascicles are grouped into, even though they are atrophying, they are grouped together. Look at the type. This is the ATPase 9.5. So type 1 will be lighter. Correct, lighter. So this is the type 1. This is the type 2. Look at the amount of atrophy that is happening in type 2 and type 1 is more. So we can call this a typical, this is what happens in SMA when there is group atrophy and hypertrophy. Remember this. They might ask you this, that I'm just giving you can remember all this. Just you can remember the words, that itself is sufficient. Spinal SMA, there's group atrophy and hypertrophy. In central core again, wherein there is, can anybody tell me? You can let this be. You can let this be. Mitochondrial myopathies, again in mitochondria, can anybody tell me what this is?
Ragged red fiber.
Yeah.
Ragged red fiber.
Yeah, this is a ragged red fiber. Correct. Oh, I think we're immunohistochemistry stains. I think this is way too high. I mean, way too much. I think this again is the one picture I showed you earlier. This is the different stains that we have. The this is again very important. Now this is the muscle dystrophies. Now we are I was asking you, where are the dystrophin proteins located in the membrane? I'm sorry, I gave you the answer. It's located in the membrane. So look at what if these are the immunohistochemistry strains. This is the normal dystrophin. This is the normal alpha sarcoglycan. Basically, you stain it for that particular protein. Okay, these are the immunohistochemical stains. You specifically ask for this. This is the reason why you need to write the classical history. You need to write whether they have whether you're suspecting a dystrophinopathy, a sarcoglycanopathy, so that they'll be able to stain it according to it. Because you are not giving them the entire body to stain, you're just giving them a small sample of muscle. Remember that. So once everything will pick up on the membrane. Okay. Now this is the normal dystrophin. Look at if it is dystrophin negative. You can let this part, you can let the H&E be. You can just look at the immunohistochemistry. This is the normal dystrophin when there is dystrophin negative. This is in case of DMD. Again, in cases of Becker's, look at the amount of dystrophin. Look at the dystrophin here. Can you make out the difference? There is complete absence in DMD, whereas in BMD, it will be patchy. Okay. Then again, for different types of dysferlin, just like I said, this is all the types of stains, like for the sarcoglycan, the alpha, beta, gamma sarcoglycans, the dystroglycans, all of them basically are immunohistochemical stains, which basically you look at the perimycin. Okay. Again, you need to look at these are all the types of stains. Again, for merosin myopathies. So I conclude. I'm really sorry. I give my sincere thanks to Department of Neuropathology in NIMHANS for helping me out with this this presentation. Again, do the right thing, but do not do everything. Any questions? I'm sorry if I went very quickly because it is a very boring topic, I know. But if you can understand how normals, if you're able to understand the basic stains, what are the basic patterns that we are able to discuss, that the basic patterns that we're able to pick up, that itself is more than sufficient. What happens in an inflammatory myositis, where the muscle atrophy will be, how the different stains appear. If somebody tells you if there is if you can't say that, you can't immunohistochemical stains, and where you should have a rough idea as to what you're dealing with, what you'll be expecting in the biopsy also, and each biopsy report that you interpret it with the pictures that I gave you according to what I said, interpret it according to it, so that you'll be able to better help out your patients. Thank you so much. Any questions?
Yeah, somebody has posted one question. So, sir, can you read and answer that?
Yeah, sir.
In the chat box.
Yeah, sir. Yeah, I think these are all. Yeah, giant axonal. What is this? Is it the same we heard as tire print in biopsy? No. I'm sorry, I didn't get to know this. Sir, nuclear and sarcoplasmic inclusions, how to differentiate? Again, nuclear inclusions and sarcoplasmic inclusions will be based on the stains that we have. Nuclear stains, again, like I said, Mason's trichrome nucleus will appear purple, whereas sarcoplasmic reticulum will have enzyme stains, will be much more useful for that. And yeah. Anything else? I'm sorry, I tried to make it as interactive and a very boring topic as interactive as possible. Shailesh sir, sorry if I couldn't ask more questions.
Sir, can you just I want to take a snapshot of
very nice presentation.
Sir. So
that recording will be sent.
Yeah, sir. Somebody
Where you
else
can
had
take-
something to say.
Anybody?
Just wanted to take a snapshot of the plus first slide. sir, in-
Recording will be
Okay,
sent.
okay,
Oh.
okay, sir.
Okay, recording will be put. Yeah.
That you can take.
Yeah, sir.
Any other student have any question they can ask?
Again, this is the last thing that they'll ask in the Viva. So when you are about to finish,
Yeah.
if you are able to answer this, I think if you can just go through this very quickly in whenever you are bored, whenever you are having food after your make sure all of you eat well after your clinical case presentation. I know all of you will be tired, all of you will be very tensed, but have some food, because by the time you come to biopsy, you'll be hypoglycemic, you'll have no energy left. That's when you make mistakes. That's the whole point of this presentation.
One more question is there in chat.
Yeah, sir. 1 second. In nerve biopsy, for selecting nerve for predominant motor neuropathy, which one to select? This is a very good question. Ideally, if we are going to select a motor neuropathy, ideally you can select the nerve which is most involved. Again, you can't create weakness, or you can't based on the electrophysiological pattern. Say, for example, you have an ulnar neuropathy, which is a mixed nerve. Ulnar is a mixed nerve. You have a motor and a sensory pattern with motor and sensory fibers in it. You pick up the based on the clinical nerve description. You don't have a stain which says this is a motor nerve, this is a sensory nerve. I hope you understand. There is no stain that says this is motor nerve or sensory nerve. It just says the nerve. But we need to know what we are interpreting. We have to pick up, when we are saying sural, we can't give a diagnosis as motor neuropathy. We know it as a sensory nerve fiber, the sensory neuropathy alone. But if we are selecting the type of nerve, based on that, we'll be able to tell. When we are selecting, for example, can anybody tell me a pure motor nerve? Can anybody tell me a pure motor nerve? Pure sensory, I told you sural. Can anybody tell me a pure motor nerve?
Peroneus tertius, we can take that now with neurobiology.
Peroneus tertius is a muscle. I asked for a nerve. Read up and tell me. This is a very this is another very worth thing. Axillary nerve, correct. Axillary nerve, correct. But axillary nerve also has axillary nerve distribution, right? And in the sensory, what is the axillary nerve sensory loss that we have? What is that called?
Regiment patch.
Correct. So you have some amount of sensory component. This is the reason why motor neuropathy, to say a motor neuropathy, it is not a biopsy evidence. This is a electrophysiological evidence. Again, this is what I said in the first beginning part of my presentation. Remember, it adds to the diagnosis. It does not make the diagnosis. Please understand, only after you do the electrophysiology, you select the nerve. Got it? Yeah, anterior intraosseous nerve, very good. But again, do you do the anterior, any indication for nerve biopsy in GBS? Again, like I said, acute GBS, there is absolutely no indication. But again, if you're going, if it is a acute presentation of a CIDP, then there is an indication to do a nerve biopsy. Again, like I said, to demonstrate acute demyelination in the presence of chronic demyelination and remyelination. That is the indication for in cases of GBS. If you do a GBS nerve biopsy, make sure you have indemnity insurance. Got it? Musculocutaneous nerve, yeah. Again, it's difficult. It takes some guts to remove the musculocutaneous nerve. Musculocutaneous nerve. That is the reason why electrophysiology is very important. Okay. Any other questions?
Yeah, in this era of genetic study.
Yeah, sir.
Biopsy.
Should we, sir? Yeah, again, that is what, sir. And biopsy plays a major role. Like I said, I had given that diagnostic utility, wherein that initial 50% it is used to differentiate in the rest 50%, to 20% it is to differentiate between two diagnoses. to specifically diagnose, like something like a Tomaculus neuropathy, HNPP, or something like that, it is helpful. Whereas to diagnose cases like CIDP, it is helpful, but not, it will just give you a pattern. If it fits to your clinical diagnosis, it is definitely helpful. But in the era of electron microscopy, in the era of genetic studies, again, remember, if you don't have a pattern, you don't know what you'll be treating. In a case of acute on CIDP, should we be expecting onion bulb pattern? Correct. We should be. If there is chronic remyelination happening, that is what I said. If there is prognosis based on if the improvement, if the patient has a very severe disease, if the patient is not improving, then we might not be able to, you'll actually be looking for onion bulb pattern in the report. That is the reason why you need to read the entire report. What they describe, what they have described in the report, you need to read. If there is onion bulbing pattern, then you will be able to have much more confidence to say that patient has chance of remyelination that is occurring. That is when your treatment is actually working. That is how you can tell the patients that, you know, you have a chance of improving. Go on with the treatment.
Yeah, if there are no further questions, we can close the session. Thank you, Dr. Suhas, sir, for
Thank you,
making
sir.
this
Thank you so much. Thank you, sir.
presentation interesting and simplifying.
Thank you, sir. Thank you so much. Thanks for
Thank
giving
you,
me this
sir.
time. Yeah.
Thank you, everyone, for participating. We can close the
Thank you, sir.
session. Thank you.
Thank you so much, sir.
Yeah.
Thank you.